Welcome, I'm James Townsend with WellWise.org here at the Natural Products Expo West. And I'm here today with Dr. Jim Josephs, who's the director of the Human Nutrition Research Center on Aging at Tufts University, who's just given a fascinating talk on what he's discovering in terms of what kinds of foods are having a dramatic effect on the aging of animals and people. Dr. Josephs, could you tell us a little bit about what you were saying about blueberries and strawberries and what you're discovering they do with mice populations and so on?
Well, the work is primarily in rats, and we have some human data which goes along with the rat data. Through our colleague Bob Kerkorian from the University of Cincinnati, he's now completed a human study, pilot study, showing similar kinds of findings that we saw in rats. We started this research to really look at what's happening during aging in the brain relative to oxidative and inflammatory stress. And basically our work and a lot of other work shows that these stressors increase as a function of age, and we wanted to see what would happen if we took some high antioxidant fruits. Like berry fruits, and we've done a number of them now: strawberries, blueberries, raspberries, thornless sweet blackberries, a whole bunch.
And walnuts.
And walnuts, yes, definitely. And we found that they were very effective in reversing some of the motor and cognitive deficits that one sees with respect to age. And we believe that these effects that we see the beneficial effects are primarily the result of their ability not only to do what we call scavenge free radicals, but they're also able to turn off these signals that the free radicals create that are deleterious to the cell and to the neurons.
And I was particularly interested in, for instance, in the motor skills, you were using what we'd call a water maze for the rats in terms of memory, knowing how to find the platform underneath the water again, and you really discovered dramatic differences between those who had a blueberry or raspberry kind of infusion, if you might say, and those who hadn't, in terms of being able to find that platform again.
Well, the blueberry and the strawberry fed animals, were able to find a platform. They were able to do it in a couple of different ways that would indicate that more than one type of memory was important, and we were affecting it, and we were having a beneficial effect on it. In other words, there's people think of memory kind of collectively, but actually there's many types. In humans, for example, the memory for names is one of the first to decline as a function of age. So we were working with the rats to see if things like spatial memory, place memory, those kinds of things were affected, and that's why we used the Morse water maze. It's nice to see the parallel here between animal and human research, because if you ask the right questions, you can get translational value from looking at animals that will actually translate to humans.
I see. And a couple of things that you said were surprising to me, and one was that the vitamin D levels are depleted with COX-2 inhibitors, such as I guess you call them the statin drugs.
Well, the statin drugs work a little. They're not really COX-2 inhibitors.
I got the terminology wrong.
But the statin drugs, that's not our work. There's actually papers on that.
I see.
They actually chelate out vitamin D. So what I found out actually talking to a guy from UCLA who's in the pharmacy school there, one of the faculty members, his name is Roger Clemens, of all things. But anyway, he doesn't play baseball. But he's the one that told me about this, and he sent me the papers, and they were there, that the vitamin D actually can be taken down to very low levels if somebody takes a statin.
We know that's true of CoQ10, and we have thought a long time that it's kind of a crime that doctors don't prescribe CoQ10 along with the statin drugs, but I had not heard that about vitamin D. And let me ask you the question. We are heavily involved at our website in nutrients and supplements in general, but your work really centers around the whole food.
That's right.
Or some of those. Did I say that correctly?
Yeah. It's kind of a it goes back to a very old saying, probably 125 years ago. It's the gestalt of the food. That the whole is more than the sum of the parts. I don't know if you were in the talk earlier today where the fellow was talking about basically the gestalt. When you look at patterns in something, the eye tends to, and the brain kind of tends to fill them in and put things in there. It also means that the whole is more than the sum of the parts. In the case of the whole berry, for example, or the whole walnut, you're actually taking advantage of not only what components are in there, but how the components go together, the ratios of one component to another. For example, the fatty acid ratio in walnuts is very important, 1 to 4.2. And that ratio is very important and actually contributes to the beneficial effects of the walnuts. When you start to take these things apart and you make for example, in the case of the blueberries, you make an anthocyanin extract, or you make this extract, you can't always be sure that that's as good as taking the whole berry. This is why I said eat the whole berry. And in fact, in some cases, in the cell models we were looking in, you actually increased the reactive oxygen species / oxidative stress when you looked at the especially in relation to putting on a stressor. It actually had a synergistic effect with it. So when you're looking at these things, it's best to eat the whole berry.
And does the in your work with the rats, do you feed them juice or the berry, or what do you work with?
The answer is yes.
Oh,
Depends
both.
on what
Yeah.
we're looking
Okay.
at.
Right.
We studied a number of years ago Concord grape juice, and we gave them the juice at different concentrations. When we studied, we've looked at pomegranate juice at one time, and gave them the juice. If it's not a juice, if we're looking at for example, a berry, what we do is grind it up and spin it down in a centrifuge, take the top portion called the supernatant, and we freeze dry that. So it comes out like a purple powder. We take that and mix it into their food. Harlan does that for us. They make lab chow.
Is that
Or
any kind of
actually
I'm sorry, is that any kind of a concentration, or is it just simply a different way of presenting the food in its natural state?
Well, we didn't want to feed berries. We wanted to make it as part of the pellet that they eat. There's a lot of waste if you try to put berries in there. It's just not a good way to do it. And you could standardize things better with this method. So we would put it into what's called an NIH 31 diet. It's a standard diet which has all the goodies in it. It's not a stripped down diet because nobody really eats a stripped down diet. We wanted to show this even superimposed on other antioxidants that are in that diet, because it's a pretty complete diet that they eat.